As life evolves, so does technology. And technologies like artificial intelligence and gene editing are the newest revolution. Personally, I think that gene editing is going to drastically change the world. If you don't know too much already, gene editing is a technology where scientists are able to change the DNA of an organism. DNA can be added, removed, or altered at particular locations in the genome. This has crazy implications. Things like sickle cell anemia and cystic fibrosis don't have a cure yet. With gene editing, a cure can be found. Along with this, designer babies can be created. Alright, before we go on, let's get into some basics. The human genome has 4 types of bases found in DNA: adenine, cytosine, guanine, and thymine. Adenine pairs with thymine, and cytosine pairs with guanine. The genome is comprised of over 3 billion of these pairs in 2 intertwining strands of DNA. DNA is first transcribed into RNA. The RNA is then translated into protein. But the thing is, misspellings of even a single letter of a gene, so A, C, T, or G, known as point mutations, can yield proteins that are dysfunctional or missing altogether, and this results in disease. So what gene therapy does is essentially replace a gene with a mutation or add a new gene to cure the disease. It replaces a faulty gene or adds a new gene in an attempt to cure that disease or improve the system. This holds promise for treating a wide range of illnesses such as cancer, cystic fibrosis, heart disease, diabetes, and AIDS. Introducing you to a company called, drumroll please, Beam Therapeutics! Sparkles, rainbows. Yep. And what they're doing is developing precision genetic medicines through base editing. Founded by lead scientists in CRISPR gene editing, Beam Therapeutics aims to develop therapies to serious medical conditions and diseases using proprietary base editing technology. By the way, this technology can make precise edits to single pair bases in DNA and RNA. Base editing is a new genomic editing technology that basically enables direct and irreversible conversion of a DNA base into another at a specific targeted genomic locus. Base editors have 2 principal components. These components are fused together and they form a single protein. So the first thing is a CRISPR protein. It's bound to a guide RNA, and that leverages the established DNA targeting ability of CRISPR, but it's modified in such a way that there are no double-strand breaks. And then the second thing is a base editing enzyme, such as a deaminase. And this deaminase, it carries out the chemical modification of the target DNA base. In this proprietary combination, what it does is enable precise targeting and editing of a single base pair of DNA. And this has never been previously done or was never possible, which is so amazing. The components for base editors can be customized, and therapeutic programs can be created for specific diseases. By changing the guide RNA portion of the CRISPR protein, what we can do is precisely retarget base editors to different genomic locations based on their gene sequences. And then by changing the deaminase, we can control which base is edited, C or A. So what we're doing is using base editing and a chemical reaction and targeting a certain DNA sequence, therefore creating a precise genetic outcome. By rewriting a single base in the genome, base editors enable a diverse toolkit, and this can be used to correct disease point mutations, modifying genes, and creating protective genetic variations. Here we're activating gene expression, silencing gene expression in some cases, and can also make multiple simultaneous edits. By rewriting a single letter, Beam has the potential to cure millions of people from serious disease. Base technology can be used in so many ways. what we can do with it is to really activate or silence gene expression, as well as cure disease that are caused by point mutations. Let's treat disease one letter at a time. Thanks for tuning in, guys. I'll see you at the next video. Bye!